Welcome to our first bioinformatics project. We're going to be making something called a Synteny plot using SV which is a R package, to make something like this example from their documentation, where we have two regions. This region is going to be our target region, which is human. This is our query region, which is going to be from mice. And we're going to be looking at something called the HOXB cluster, which is a very evolutionarily conserved gene cluster in vertebrates that assigns the architecture of early embryonic development. So this is going to be a helpful project. We're going to learn a lot of different tools. Definitely follow along as you're doing this. I want to say that I'm going to be working on a Windows PC. If you're using Mac, you definitely want to find analogous commands as necessary. And a lot of them are going to be the same. So I'm going to be working in the Ubuntu Windows subsystem for Linux shell, and that's going to have a lot of the same commands as the Unix commands for Mac. But if you run into any trouble, ask ChatGPT or Claude for advice for a specific command. But I think we should be all set to go. There's going to be a lot to cover. It'll be cool. We're going to start by looking at the actual genomic locations using something called IGV. And we can download IGV for free. It's an integrated genomics viewer. And we're going to go ahead and go to download the desktop application. Once that downloads, we'll be able to look at the actual cluster. And the reason we're making a Synteny plot is because Synteny plots allow you to see the shared regions for genomic loci. What that means is you can literally look at the same genes in two very related strains of the same species, such as an agouti mouse and a black mouse with different genetic backgrounds, or the same loci in different species, which is what we're doing when we look at the same cluster of the same genes, or basically orthologous genes, in a human and a mouse. So it'll be really cool. Let me go ahead and figure out where I want to put IGV. For myself, I'm going to go ahead and put it on the C drive for me, and I'm going to make a new folder called IGV. So I'm going to go ahead and install it right there. It'll take a second to install. What's nice about IGV is it's a robust genomics viewer that's used standard across bioinformatics. So it's not like professional scientists are using something way different. Literally, I use this at my job where I do molecular and bioinformatics research full time. So literally, I'm in IGV, I'm doing this stuff. It may not open automatically, so if it doesn't, we can go ahead and go over to the IGV folder. And sometimes it takes a second to identify the correct one. In this case, it's IGV launcher, and that should get us up and running. It does. Fantastic. To orient ourselves, we can see a few things. So we're looking at the human genome right now. This is the HG38 version, which is a pretty current version. Down at the bottom, we see the actual gene annotations, which will become a lot more clear once we start zooming in. So for example, we can go up here to chromosome 1, and here at the bottom we see all of the different genes on the human chromosome 1. We can also see up here the actual length of this region, which is 248 MB. MB stands for megabases, where one base is literally a base pair of DNA. And it might seem kind of abstract, but we can literally zoom in a lot. And once we zoom in all the way, we'll actually see the actual DNA sequence. And I think, make this a little bit bigger. Let me see if I can do that. If I can't do it easily, then I'll just go from there. Oh yeah, I think that might work. Okay, that didn't work, but basically you can see here that the actual DNA sequence is down here on the human genome. DNA, as we know, is 4 nucleotide letters. So we have T for thymine, C for cytosine, G for guanine, A for adenine. And we can see GACCT, CCTT. So literally, you can zoom in and see the actual DNA sequence, which is pretty awesome. We're going to be looking at a specific cluster of genes in both human and mouse, which is the HOXB cluster. Like I said, this is a very conserved, very ancient gene cluster in vertebrates. And we can see, let me do that a little slower. So we have HOXB1 right here. And what's cool is as we move over a little bit, we can start to see other HOXB genes, like here's HOXB2, HOXB3, and if we zoom out even more, we can see the entire cluster. I think this is a good view right here, where we see HOXB1 over here on the 5' end, and then all the way over here, HOXB9, and we go HOXB1, 2, 3, 4, 5, 6, 7, 8, 9. So we have a group of 9 HOXB genes here in this cluster. Let me zoom out, and we can see next to it we have other genes. We're going to be focused on these 9 HOXB genes today. So we'll make a note of this. We're actually going to go ahead and also look at the same cluster in mice. To do that, we'll go to Genomes, download host genome. We'll go to mouse. MM39 and MM10 are both more recent versions, and they're both really good. We're going to look at MM39 today. So we can go ahead and click MM39, click OK. It takes a second to populate. Once it does, we can go anywhere, like chromosome 1 again. This is in mice this time and not human. And we can start looking at mice genes. For us, we want to look at the HOXB cluster in mice as well. So we'll go ahead and type HOXB1, and we can see HOXB1 is right here. And as we zoom out, we can see the entire cluster. And notice it goes in a different order than it did in humans. So that might be an interesting thing to see in our Synteny plot. So I can zoom out one more time. And now we see we're going from HOXB1 all the way to HOXB9. So we're going to need to know these genomic locations, because what we're going to do for our pipeline is we're going to cut these regions in human and mice, and we're going to use a tool called SAMtools FadeX to do that. That's going to feed further into our pipeline. We're going to need a few things for that. On Windows machines, I think the easiest way to do it is to use Windows Subsystem for Linux for all our commands and for downloading our packages. So if you don't have it installed, you want to go ahead and install Windows Subsystem for Linux. And let me see here if we already have it installed. Okay, so I think we're all set here on my machine. I was just wondering if I had installed Windows Subsystem for Linux on this one yet. Looks like I have. If you haven't yet, it's very straightforward to do so. You can just type install Windows Subsystem for Linux, and then it can walk you through how to do that here. Very straightforward. We're going to go ahead and go from here where I already have it installed. We're going to need 2 packages here. We're going to need SAMtools, which is a very standard bioinformatics setup. So SAMtools is written in C, and straight from their GitHub, I can say that it is literally like just a straight up established useful tool. It's been running for more than 12 years, and it's just very useful. I think the best way to say it is, actually this description here. Set of utilities for interacting with post-processing short DNA sequences. To get it up and running, we're going to go ahead and do sudo apt update. You can type your Windows Subsystem for Linux password. That's going to just refresh some things, because we're going to need to have a C compiler ready to go to be able to use SAMtools. Sudo apt, I think, let's see, apt install build tools. I think that's it. Yeah. Unable to locate package build tools. Well, let me go ahead actually and first try to install SAMTools, see if I need any other dependencies. Because if that works, then that's, okay, great. So that did work. So once you get to Windows Subsystem for Linux, the two things you want to do for sure are sudo apt update and then sudo apt install SAMTools. That's going to get our SAMTools up and running, and we can test that it's working by typing SAMTools, pressing enter. You do want to see a list of helpful commands for SAMTools ready to go. And we're going to be using one called SAMTools fadex. And you know it's working if it gives you some help on different options for that. Fantastic, that's working. Now we need last Z, which is a setup here from Penn State. It's a program for aligning DNA sequences. It's a pairwise aligner, especially useful for different species. If you want to look at sequences of DNA from the same species, just different strains, you want to use something called Minimap2, which is a very powerful aligner for close species. But we're going to be looking at last Z today. To do that, we're going to do sudo apt install last Z. And then that should be good to go. You'll know it's working if you type last Z and it gives you a set of useful commands, which that did work. Perfect. So we should be good to go on that front. I'm going to go ahead now and open up some boilerplate code I have for getting the FASTA regions, and I'm going to zoom in a little bit. This is a little bit easier to zoom in on. We're going to need to go back to IGV to actually find the locations we're interested in looking at. Because, again, what we need to do is excerpt the actual cluster of the HOXB genes. So we can go to HOXB1. And to do that, the syntax works like this. You want to feed SAMTools something like this format here, where we're going to do SAMTools fadex, and then we're going to give it our genomes files, which are FASTA files, .fa for human and mouse. So we'll go ahead and do that. And then we're going to give it a sequence of characters for where we're going to actually extract from. And that's going to be typed where we're going to do basically chromosome 7, start and end locations. And then we're going to specify where we want the output file to go and what we want it to be named. And we can actually assemble it here so it's ready to go. So we're going to go to IGV. Notice here the syntax is chromosome, colon, start to dash end location. That's the same syntax SAMTools fadex uses. So we're literally going to go ahead and take this setup right here, because this is the range of the view that we have now. And you can see that that's true, because if we zoom out, we see that this sequence is changing, where the start and end locations are changing as we move across the view. So if we literally want to make sure that we capture things in the HOXB cluster, we can go to where we feel like our view is currently capturing the entire cluster, which I think this is doing a good job at capturing that now. And we will literally just copy and paste this so we're ready to go. Awesome. That's going to be our start and end locations for SAMTools fadex. Now we need to get the FASTA genome files for human and mouse. This is just for human. We'll do a separate one for mouse. To do that, we want to go to the UCSC genome browser, HG38 download. We'll go human, we'll go HG38. And then it's actually so simple. Let me say too, if you're confused what these files mean and what this is, UCSC genome browser is one of these standard genome repositories anywhere. It's used widely by biologists, bioinformaticians, so it's really good. Here it gives you metadata on what each of these files are. We're going to be using this one, hg38.fa.gz. To orient ourselves, this is the human genome in FASTA format, zipped via gzip. It's a soft masked assembly sequence in one file. We're going to take this one and get it into our setup. So let me go ahead and I'm going to right click on the one we want, which is this one. It's 938 megabytes. Right click and then copy link address. We're going to go back to Windows Subsystem for Linux. And I'm actually going to move first into the file folder that I want to be working in. And I know which one that is. If you want to move into that folder, you type CD. And I'm actually going to move into my main setup right now. Let me see, actually, what is the username here? It's DG1000. Okay, and then documents. And then I'm going to go to, oh, hang on. Yeah, so for me, I want to go actually, okay, cool. I was typing a few things because I wanted to orient where I actually want to put this file. So now I'm in the correct folder I want to be in, which in my case is called YT, the underscore Synteny HOXB setup. So I'm going to do something called wget. I'm going to type or copy the link that I just got and then press enter. That's going to start downloading the genome file for human HG38 into the correct folder that I want. So for you, the correct folder is going to be different. It's really anywhere you want on your computer to be working in. This is where I want to be working in. While that's downloading, I'm going to go ahead and grab the link for the MM39, which is really easy to get. It's going to be this one, mm39.fa.gz. We're going to go down here. We're going to grab that one, do the same, copy link address. And you can confirm, like, this is the mouse genome MM39, which is the one we were looking at here in IGV, which is perfect. We're going to be grabbing that HOXB1 setup along with all the other HOXB genes from here for sure. So that's perfect. We'll go ahead and once this one is done, it's at 75%. Once this one's done, we're going to wget that genome as well. And you'll hear my computer fan probably going off. It's just doing its best and it does a good job. So this is, as this is running, let me explain as well why we would want to do a Synteny plot. Synteny plots are nice because they can show you where the sequences are conserved for clusters across genomes. And here we already know before doing this analysis that the HOXB cluster is very conserved. The whole cluster and all the genes in it are very conserved in vertebrates. So it just ends up being a useful tool for us, and it's good practice for bioinformatics. We're going to do wget and then get the mouse MM39 genome, which is great. While that's running, we can go back into IGV. We can get the HOXB cluster in our view the way that we want. And let's say, yeah, so we're getting close to it. This is a little bit too much on the ends. And if you can't quite get the view that you want, the best way to do it is to get enough to where the ends are in the right location you want, and then a little extra. What I mean by that is, like, literally, we'll go to HOXB9, and I'll add about like 10,000 base pairs. That's going to be our start location. So I'll go ahead and copy that into our notepad file over here. And let me just make a note, this is HG38. And then down here we have MM39. Oh, 39. sorry, so this is our start, and our end location, it's going to be over here. So the end of HOXB 1 is right here. We'll add a little bit of buffer, like 10,000 base pairs. So that's going to be this value right here. So this region is going to be our HOXB cluster in MM39. This is great. So we have both genomes now ready to go, and they're zipped. So we want to unzip them. You can do gunzip star.gz. That will unzip both of our FASTA files right now. It might take a second because they're both about a gigabyte. So it's not a small amount of data to unzip. Once those are unzipped, we'll take a look at the start of each of those files and see if it looks good. We'll see also the format of what a FASTA file actually looks like. So we want to make sure this is set up. And let me go back. Yep, so this is still unzipping. So I'll go ahead in the meantime and start assembling our samtools fadex commands. So this is super straightforward. We're going to put samtools fadex. We're going to specify where the genome file is. Genome file, I'll just write genome file for now. We have chromosome. In fact, I'll specify this as HG38. And then our output, so we're going to do -O. We're going to put // because we want it in the current directory. And we'll go ahead and say this is going to be called HG38 HOXB cluster region .fa. .fa is just going to be specifying it's a FASTA format. We can do the same thing down here. So we'll put samtools fadex, and then we're going to do MM39 genome file. Of course, we're going to change that to the actual genome file name, and then we'll put MM39 HOXB cluster region.fa. Okay, let's see. So those are both unzipped now, and we can take a look at HG38.fa, just the head of that. It's a lot of ends. That totally makes sense because it's just not all of the chromosome, not all of the locations on this setup are known, or it might vary depending on the sequencing. So N is totally expected. N is not a nucleotide base necessarily, but we can go ahead and go to the end. That might be more illuminating. Okay, this is great because this is the end of the human genome file, and we can see that what we expect to see, ACs, Ts, and Gs are present, which is great. We can also do the same with MM39. Head, okay, yeah, but what if we go to tail, MM39? Okay, lots of ACs, TGs, so, and the fact that it's different as well means that it didn't somehow duplicate our files or anything, so looks like we're good to go on that front. So now we want to make sure we get the actual genome file names correct. And we can see it's just simply HG38.fa and then MM39.fa. So that's looking pretty good to go. And let me go ahead and, yeah, sure, just say that. Now we're going to go ahead and do our first attempt at using samtools fadex. If it gives any errors, we'll address those, but let's just check it out. So we're going to copy that command, and we're going to go ahead and paste that and run it right now. Let's see if it works. Let's see if it gives us any issues. It'll tell us if there's anything wrong going on. And the speed at which this executes depends on how much RAM, CPU power your machine has. So that might take a few minutes. As that runs, I'll go ahead and start getting us set up for the next step, which is going to be last Z. And actually that did run. Let me go ahead then and run the samtools and then explain last Z. So that's running the mouse samtools fadex. Our last Z command is going to look like this, where, like I said, last Z is a pairwise comparison tool for different species for the same genomic locus. So we're going to look at MM39 and then not rat. This is just an example format. We're going to be looking at the setup for human and mouse on their respective chromosomes. Oh, I should have mentioned this earlier. We can see even now that the same locus is on different chromosome numbers. So in human, it's in chromosome 17. In mouse, it's in chromosome 11, which is already kind of cool and interesting. The way that last Z commands work is you want to put the target organism first and then the query organism second. And we can see that even in the last Z help documentation. So it will tell you usage is last Z, target, and then query, and then different options. In our case, the target is going to be human, and then the query is going to be mouse. We're going to go ahead then and make sure that we get the names of the correct region files correct, and then specify path format, and then go from there. So now that the SAM tools is done running, I'm going to do list all our files, and we have the correct cluster region set up. So I'm going to literally just copy these names here. So this is going to be last Z HG38 HOXB cluster dot fa, and then we're going to do MM39 HOXB 30 or HOXB1, sorry, cluster dot fa. Format is path. Those are just the format files we use. And then we're going to name this a useful name for us. I think the best sort of name for this would be query mouse MM39 chromosome 11 HOXB1 HOXB actually HOXB cluster target human HG38 chromosome 17 HOXB cluster last Z.path. It's kind of a lot, but we'll know even years from now what exactly this file is. So we'll go ahead and try copying this in, see if it gives us any issues, address any of those issues, and then go from there. So let's go ahead and see if it actually outputted a file. If it did, it would output it here in our specified folder. It looks like there is something, so we can go ahead and look at that file right now. So we're going to go nano and then open that file. And this is really good because this is what we want. We want to see the different setups on each side. So we have, let me go ahead and go back. To orient us, the query is going to be mouse chromosome 11, target is human chromosome 17, and we can see chromosome 11 here, chromosome 17, and then we have a few different numbers as well, which is really good, and this is going to make more sense as we actually plot it, because this information is going to get fed right into our R script to make the actual figure. So we'll go ahead and feed this into RStudio. If you don't have RStudio, you want to go ahead and download it. You'll need both RStudio Desktop and R. So I actually don't know if I have R on this machine. I think I don't. So I'm going to download both R and RStudio. So I'm going to go ahead and install R for Windows and then RStudio. Those are both installing right now. As those get going, we should be good to go for the next step. I'm currently installing R and RStudio for the first time on this machine. Let me go ahead and do R first, and then we'll go from there. So you want to just accept everything. R and RStudio are both free, so it's really nice, really straightforward. That's working pretty good. And as that's installing, let me go ahead and orient ourselves to what we have so far. What we've done so far is we've gone into IGV to look at the genomic regions of the HOXB cluster. We've installed SAM tools and LastZ to our Windows subsystem for Linux setup. We've used both of those tools to first use SAM tools FadeX to slice the actual cluster region for the HOXB genes in both human and mouse genomes, and then we used LastZ to create a path file, which is basically a file format that's going to be useful for creating the plots using the R package SV by I. We're currently installing R and RStudio to this machine. So R is installed. Let me go ahead now and install RStudio. And it might sound redundant, like why do we need both R and RStudio? It's really because they're actually different things. R is the actual language itself, the programming language, and then RStudio is a graphical user interface. Specifically, it's an IDE GUI, so like a integrated development environment, graphical user interface that's useful and really nice for using the R programming language. So it's really good. It's one of the standard ways to use R, but it's not the only way. You can, for example, use Jupyter notebooks with a R kernel to code in R. You don't need to use RStudio, but you do need R, the language itself, to be able to use RStudio. Now that the setup's finished, I think it should be good to go now. Let's see, RStudio. Let's go ahead and try opening it, and we will go from there. Okay, I think this is working, and it looks like I opened two things. We're gonna start a new project. Doesn't necessarily need to be a project, you just need to get a script going. I'm gonna go ahead and open it in the specific directory I want. Okay, let me go ahead and see if that worked. If it wants to allow anything, just go ahead and allow. Oh, interesting. LS is not recognized. It's okay. So I think that actually makes sense. It's using the Windows thing. But we will go ahead then and start a new R script here. I have a boilerplate ready to go for us that's going to have the R stuff. So we will go ahead and open that up right now. Let me go ahead and do that. Azure Synteny boilerplate. I'm opening Visual Studio code right now. You don't need it. You can literally just use RStudio directly. I just opened it real quick because I'm just going to copy it in here. Okay, cool. So it's copied in here, and what I'm going to do is I'm going to go ahead and save this. Save this one as HOXB issue 38 mm39 Synteny. Okay, cool. So to orient ourselves here in this file, we first want to make sure we set the working directory. We need to install all these packages in R as well. We'll read in the path file. We'll read in annotations later. We won't worry about that yet. And then we'll annotate the files, and then we'll go from there. So let me go ahead first and install any R packages we need to. We'll need the ones read R, dplyr, sb by I, paffer, stringer, genomic ranges. So I'll go one by one. Some may need to be installed. So the best way to do that, and it sometimes varies a lot, so I'm literally going to try install read R. It may or may not work. Okay, cool. So what I'm going to do is I'm actually going to refresh myself on how to go ahead and install these packages. Real quick, just a quick search will remind us how to do it. So install packages. So let's see if that works. Install packages. And sometimes it works, sometimes it doesn't. Okay, that seems to be working. It wants our tools. Install packages. Okay, cool. Please download and install the appropriate. Okay, so it wants our tools. That's cool with me. Our tools. So it wants us to install our tools, which is totally great. And to do that, I'm going to go ahead and do it the way it wants. Our tools 45 installer. Let's see if that works okay. I think that should work fine. And this is what you run into as you're working on these projects. Sometimes you need to just try stuff out. Any errors it gives you, don't ignore them. Don't fall into despair. Just do what it asks you to do. Oftentimes it'll just tell you why the error is the way it is, and you just have to figure out how to solve it. Use Google, use resources like ChatGPT and Claude to help you along the way. Like this is literally just saying we just don't have readr, and that is telling us, okay, we need to go ahead and install that. This warning is telling us it wants our tools, so we're installing our tools right now, and that's super chill. Let me see, where is our tools? Okay, it is almost done installing right now. So I'll go ahead and go over to downloads. Our tools, let me go ahead and install that. Oh, that's kind of large, 471 megabytes, wow. Okay, but it's fine, let's do it. Let's do it. I'll actually close our studio for now because generally it's gonna need to restart anyways to get our tools running. Awesome, our tools is currently installing right now. One thing I hope that this illustrates is that there actually are a number of steps that go into creating even a single figure, because the output of all of this work will be one figure. And I hope that helps all of us to appreciate some of the complex, sophisticated figures we see in scientific publications, not only in biology, but in physics and other fields, because there is a lot of work that goes into even a single figure, because we're having to install lots of different packages and tools, and it's totally worth it. And it is really cool, but almost like art. Someone who is unfamiliar with it may not be able to appreciate the level of work that goes into the output at the end of it. So that's just some context there. And it looks like our tools now should be all set to go. We're gonna go ahead and try opening our studio again. Our studio is nice because it often goes straight back to where we were. So let me go ahead and try a few things. So, okay, library. It's good that readr is loading because we did previously try to install it. Now it's running. If all is correct, then this actually will give us an error because it shouldn't be installed right now. So we can go ahead and install that one now. We'll go ahead and do dplyr. This is very useful for reading and manipulating data frames in R. It's kind of analogous to pandas, which is the really good Python data frame library. So it looks like that worked. We'll go ahead now and install svbio. svbio usually has a number of different dependencies, so it'll probably give us an error, and we'll probably have to, yeah, okay, so it looks like it didn't give us an error, but it gave us a warning. This package is not available for this version of R. A version of this package for your version of R might be available elsewhere. And let's, because we do need to use this package, so we might have to actually downgrade the version of R that we're using. Let's go ahead and go to svbio and just take a look at the versions that we need. Let's see, references, svbio. So this is just what real-time figuring out what things work. So it looks like 4.3 should work. Let's see, install packages, dev tools, install required packages, dev tools. All right, let's try this. We're gonna try doing this exact setup right here. We'll try it. So install packages, dev tools. And this may or may not work. And just to orient myself, I'm literally at the svbio GitHub repository right now. I'm trying their instructions in their README file to see if this will work for us. So it looks like that worked, and we'll check that the library we just installed is working okay. Okay, now we'll try installing from the repository. This may or may not work. And if it doesn't work, we will go ahead and downgrade our version of R so that we can use SV by I. And as that's going, I'll just say too, like all these steps are standard. Like it's not like the pipeline for something new like this on a new computer always works seamlessly without any dependency issues or anything. You do have to figure out things and troubleshoot to get the final result to work. It looks to me like that worked. We'll know if we type library SV by I and it works. It looks like it's installing some other dependencies as well. So we'll let that just go. And if you're newer to programming and you're wondering like, why are we even doing this? The reason we use packages and dependencies is that really packages and other packages that they depend on are built by people to do specific functions, to do specific things. In this case, SV by I is a really useful, specifically made package for structural variation among whole genome assemblies and then even regions of genomes, which is what we're doing right now. And there are other packages that do this, but this is just the one that I'm most familiar with, and it's very useful for our specific purpose. And as I'm scrolling, we can see a lot of different use cases where even with the same data set, you can look at specific regions of data, you can color things in different ways, you can look across not just two, but many different strains or species and customize the data output that you're looking at. So it's pretty nice. And it relies on other packages from other people who are unrelated to the people who created SV by I, like David. So that's just how these things go. This is just normal stuff. So let's try loading this SV by I library. If it gives us any issues, we will work within those constraints. We'll downgrade R if we need to, that's okay. But fortunately, it looks like it just worked, which is great. So we'll go ahead and keep trying. So has no library Paffer, that's okay. We'll go ahead and install Paffer using this syntax right here. So we have Paffer and that, okay, so it looks like this one is not available for this version of R. Let me see. I think we do need Paffer. Let me go ahead and go to their GitHub and see what to do. Paffer R. So we have Paffer. Let's see if they have any helpful tools for us. So we have DevTools, install GitHub, dwinter, Paffer. Okay, let's see if this works for us. This may or may not work. Because if it does, then we are set to go with this one. But we will see. Okay, now we can go back and try to run this and see what it says. Loading required package. Okay, cool. So that actually did work for us. Stringer is just worked. That's great. Okay, now we'll see. Library genomic packages. Apache package generics. It looks like we're set now with the dependencies. Now let's try setting the working directory. Cannot change working directory. Oh, interesting. Well, I actually wonder then, can it just read, because we might not actually need to manually set the working directory. In fact, yeah, let's go ahead and take a look at our files here. We have our path file here. So I think we can go ahead and take a look here. ls, great. So we'll go ahead and try reading this path file now, and that's going to be a necessary step for us to be able to move forward. So we'll, oh, that did not quite work. Let's try that again. It may or may not just copy cleanly. Okay, that did not work, but fortunately, it's not that much to type. So let me just type that out real quick. So basically I want to make sure that everything is typed correctly because case matters. So it is case sensitive. You want to make sure that everything is exactly how it's set up in the file name. But it's not too long, so it's not insane to type it out. Okay, so that's our path file. This is just boilerplate from a previous project of mine, so I'm going to rename it something more useful for us. And in fact, I'm actually going to change all instances of dog human path to mouse human HOXB path. Cool, that's great. So now let me go ahead and try reading it in. Any issues? So, okay, what we expect to see from this variable, data variable we have named mouse human HOXB path, is the same exact data here, except it's formatted in the way that the rest of our R script wants. So let's go ahead and try opening this and see what it looks like. It looks like it worked great. We have everything set up exactly the same way here, except it's read in as a data frame, which is perfect. So it looks like we're good to go on that front. I'll say too, we noticed something we already saw in IGV, which is a lot of the strands for the HOXB genes are aligned in the opposite direction. Remember how in human, we saw that the HOXB genes go from left to right. So we can see HOXB1. HOXB1 to HOXB9, it goes 1, 2, 3, 4, 5, 6, 7, 8, 9. But in mouse, it goes from right to left. We can go back to HOXB1. And we can see HOXB1 is on the right, HOXB2, 3, 4, 5, 6, 7, 8, 9, right to left. And that makes sense that the strand direction is opposite, or minus in this case. And that I predict will be interesting for us as we look at the file that's outputted from this. Let's go ahead now and turn to doing some data formatting and renaming of our columns. Here in our data frame, we have q.name. This is query.name, query length, start end, strand, target name, etc., etc. We're going to rename the column names for query and target a little bit more informatively. So instead of doing q.name, t.name, we're going to rename q.name to the query, which is mouse, chromosome. And we, see, it's nice. We don't have to check any other files like, oh, what chromosome was it again? It's literally in our file name. So it's literally chromosome 11. And we can just confirm that by quickly going back to IGV. And we see up here, chromosome 11. And then what chromosome are we at in human? Well, we're at HG38 chromosome 17. So we'll rename that like that. And we'll go ahead and run both of those lines here. And if we take a look again at that file, we can see that, oh, I should have clarified. Yeah, it's actually not changing the column name. It's actually changing the value to something more informative for us, which downstream will be nice. We're gonna not worry about GTF files right now. Those are gene annotation files. I'm gonna come back to that after we do our first draft of the plot. Same with the annotation stuff here. We're not gonna worry about that right now. Okay, so now we're gonna do some things where we're gonna change basically this placeholder to something more helpful for us. So that is gonna be HOXB cluster. We're gonna go ahead and replace all of those right now. So HOXB cluster list is gonna list the path file. This is useful if there's multiple path files where you're not just comparing two species, but you're comparing like 7 or more or less, but just more than 2. For us, it's fine. So we're gonna run that. We're gonna run this one. Oh yeah, bind rows, that makes sense. There's no rows to bind, so it makes sense that it's not gonna do that. So then instead of that, we're gonna go ahead and, let's see, in fact, hang on, let me see. List of 1, Yeah, actually we don't need that now that I think about it. List is only useful if you're gonna be using multiple path files. And we're gonna ignore the annotation for now. Okay, so we're gonna go ahead and take this setup right here. We're gonna go ahead and let's see, cluster ano. We actually don't need this right now, so let me go ahead and, in fact, let me go ahead and do up here. I'm gonna not comment that actually, because I'm gonna come back to that later. Okay, now we are gonna use this one. So this is not gonna be human chromosome 7, it's gonna be human chromosome 17, mouse chromosome 11. Basically, we want the order of the plot to correspond to target, which is human chromosome 17, and then query, which is mouse chromosome 11. Okay, let's take a look then at our individual path file, which in our case is going to be mouse human HOXB path. Double check the data, that's looking good. So we're literally going to do one thing real quick. We're going to do HOXB. I know it wants this one, cluster, HOXB cluster paths. Yeah, I'm literally just assigning this variable. I'm copying our single path file into this new data frame because the way this boilerplate is, it expects to see a list of different paths that are assembled into one larger path file. Here, it's only one, but I already just know it wants it this way, so I'm gonna go ahead and do that. So I'm gonna run this code, which is basically gonna plot our path file, and it's gonna color by direction. We're gonna adjust this color palette to whatever we want, but that already should work. Let's see if it gives us any issues. We're gonna try plotting this. In fact, before I actually show us the plot, let me come over here because we see that I created this HOXB cluster dir, which is a large ggplot object. That's promising because our plot is going to be a ggplot plot. It sounds redundant, but literally ggplot is the package name. The plot itself is going to be outputted as a ggplot object. If all worked well, when I run this line now, it should output a plot down here. So I'm actually going to optimistically resize our window, expecting to see a nice plot. If there's any issues, we will work with those. Let me click run, and then we'll see what happened. Okay, this is awesome because what this is literally showing us is everything we expect to see. So we have human chromosome 17 up here, mouse chromosome 11 down here, and we see that the alignment direction is entirely opposite. What I mean by that is there's no gene, or there's really, I don't want to say gene actually here, there's no sequence of DNA that is entirely maintaining the same orientation. It's all flipped. Basically, this whole HOXB cluster is flipped, which is literally what we saw in IGV. Again, this is mouse. It goes from right to left, HOXB 1 to HOXB 9. And we go back to a human, it's going left to right, HOXB 1 to HOXB 9. So we literally expected, if this worked the right way, for the entire cluster to be flipped. And that's exactly what we see in this plot, which is really awesome. So that did work for us. Fantastic. And we can do a lot more. One thing I'll do just real quickly is show that you don't just have to color by direction, you can color by identity. Identity is literally going to show you the values in terms of the percent identity across sequences. So it looks like here, all of these sequences are below 90% identical, but they're still pretty good because we wouldn't expect them to be 100% identical because they are different species, but the HOXB cluster is very conserved. So for us, it's still good. And we can do more work downstream to adjust these values so that we can actually break it down into the specific percent identity. So I won't worry about that right now, but basically we're gonna stick with the direction actually, because I think that's more informative for us. So let's go ahead and rerun these real quick. So yeah, direction set up right here. And if we want, since this is all the same color, like we can literally adjust the color of the minus to anything we want. Like let's say we go over to color palette. I think it's cooler. Oh, it's this one. Yeah. So just find a color you want. Anything you want is cool. Like I can literally pick a blue or a purple, like anything you want. It's cool. I'll just do this one. Let's just try this. See if it looks good. And then we'll see that, yeah, the color changed. So yeah, I mean, literally whatever you want. Like for me, my favorite color is cerulean. So I'll probably just go ahead and grab that. Cerulean. In fact, let me go back to coolers. It might have cerulean. And this is just for fun. Like you don't have to do it this way. Like for me, I might go explore palettes, cerulean. I don't know if cerulean will pop up. I literally don't know. That kind of looks like cerulean, kind of. Something like that. Maybe more like, yeah, I'll grab that one. No, that's a little too bright. I think actually, let's go this one. Okay, I like this one. So this is blue. It's showing that for this whole cluster, the entire HOXB cluster is flipped in human and mouse. That's really interesting and really cool. And now we can go ahead and add some gene annotations to make it a little clearer for us what's actually going on. So if we go over to, if we head over, let me see, to up here, we can see that there's spaces to put gene annotations. And what that means is we can input files that look a lot like this right here. This is the boilerplate for a gene annotation. And this is just the established format for gene annotation files, which are GTF files. We have chromosome, number, metadata, start, end locations, more metadata. This is positive or negative, and then more gene metadata like gene name, gene ID. We're gonna manually create these actually for ours. Why are we gonna do that? It's literally because we want to specify where the genes are of the HOXB cluster on our locus size. Let me do one thing real quick. I'm gonna name this HE38 HOXB cluster .gtf, something like that. Okay, and then I'm gonna go back to this. Let me say one thing as well, that it is interesting that the range of the cluster size is very similar. These are like the same size basically, but that's not always guaranteed. The cluster might have been very different sizes, but it is interesting that in humans, the cluster is about 100,000 base pairs long. In mice, it's about 100,000 base pairs long. So these are basically the same size. That's pretty interesting. What we're gonna do is we're gonna add rectangles to specify where the genes are, and we're gonna try to give them informative labels as well. So we're gonna do that right now. To do that, we're gonna create manual versions of GTF files. You don't actually have to create a manual version because there are gene annotation files ready to go on the UCSC genome browser website. Here in genes, we can literally download the whole set of all the genes in mouse and human. But the thing is, we don't want all the genes. We actually just want specific genes. And we want a specific way to display them. In IGV, for example, if you look at HOXB2 as an example, we'll see the introns and the exons here, right? We actually want the start end location to be one rectangle on this plot. We don't necessarily need to know the introns, exons right now, although that's useful. For our purposes, we just need the rectangle to show the span of the gene. So to do that, we are going to start over here with HOXB1, and we're going to include that here in our metadata for it. What does that actually look like in practice? Well, we're literally going to go in here, and we're going to start typing where this gene is here. So we're going to do chromosome 17. And for our purposes, this metadata isn't as important. It's tab separated, so you want to tab every time you're going to add a new piece of data. And following this format up here, we're literally going to do, and I can zoom out just to show like, show this setup here. So we have chromosome 1. Okay, right. So to simulate this down here, okay, hang on, let me do one thing real quick. Okay, I was hoping that would be informative. That was not. Okay, but we're gonna do chromosome 17 ... start location, tab, end location, tab. We'll just put dot. We'll put minus because it's minus here, and then we'll put dot. Here it is important that we put gene name, and we're gonna copy the name here. HOXB1, semicolon. We'll delete this first line because that was just as an example. And that is HOXB1. We're gonna manually do that for all the other genes. There are automated ways to do this, but this, it's not, since it's only 9 genes, I feel like it's not a lot of work to do manually. And it also illustrates what's happening. I think it's a learning experience to see it manually done. So we'll keep doing it manually. So it's very simple. Chromosome 17, start location of HOXB2, end location of HOXB2. We'll just put a dot, minus dot, gene name, HOXB2. Okay, we'll move on to HOXB3. HOXB3 is here. We're gonna go ahead and copy the stuff as well. Chromosome 17, tab, and then start location, end location, minus dot, gene name, HOXB3. Okay, cool. Let's go ahead, and it just rinse and repeat. HOXB4. Looks like HOXB3 is definitely longer, so we'll go HOXB4 over here. And in the case where there's, oh, actually no, these are both different, so that is helpful. Sometimes the same gene has multiple annotations. You may want to use all or one, depending on your purpose. For us, we'll just pick one to try to get the whole span, because that's really what we're interested in, seeing the visual span accurately across the genome. HOXB5, and there's only 9, so we'll do all 9. Oh, gene name. And you do want to make sure that you spell everything correctly, because it does matter in the code. Coding languages are generally unforgiving about misspellings or case. Like, you want to make sure that the uppercase is correct, lowercase is correct as necessary. For here, if I put instead of this like gene name like this, that will not work for downstream stuff. So we always want to make sure that it looks correct. Okay, so that's HOXB6, HOXB7, chromosome 17. Okay, we're almost done with human. Then we'll move on to mouse. HOXB8. Okay, we're almost done. HOXB9. And all the things I'm doing, like there are actually different ways to go about this. You could, for example, use UCSC Genome Browser to get these. You don't necessarily have to use IGV to get these locations. It's kind of how you want to do it. For me, I find that just doing this way just works. Like it always works. It's quick, especially with just a few genes. So I find that it's totally fine. So I saved that over here in the HOXB cluster for HG38. So now moving on to MM39. So we'll go ahead and do the same process in MM39. And again, I hope this illustrates that it's cool because you have to use a lot of different tools sometimes to create figures and to draw analyses. And again, I hope it illustrates like how cool and how much work can go into even a single plot. It's not trivial necessarily to do this. It's not like the hardest thing or anything, but it does take some work. So you do have to think about how best to do things, you know? Okay, gene name, HOXB1, and I'll lowercase it because the case does matter for gene names. That's already specifying it's not human, but it is mouse. And I deleted that first line because that was just a placeholder as an example for us for human or from the human file. Okay, let's keep going. Chromosome 11. Okay. Okay, HOXB2. Okay, we have a few things going on in the HOXB locus for mouse. I'm going to make an executive decision to go ahead and go straight to HOXB3, and that will be enough for our purposes for today. We could get more gene annotations in here, but I think that's fine for us. Plus gene name HOXB3. Okay, and then let's get HOXB4. HOXB4. We have this and this. Okay, awesome. HOXB4, HOXB5 right here. Yep, then you just, gene name, sorry. Okay, HOXB5, HOXB6. Okay. And we're almost done. HOXB7. Okay, two more. HOXB8. I find that this can be pretty relaxing. You can even put on music and just vibe and type it out. Especially once you get into a rhythm and you know exactly what you need to do. You can just hunker down, execute it, and then get it done. It's pretty cool. All right, so this is our last one, HOXB9. Okay, awesome. That's good to go. Let's go ahead and go back into RStudio. Let's take a look at our file list. So we have these here. So we will go ahead and grab our files. So for human HG38, we're gonna type HG38 HOX. Oh wait, what did I name it actually? HG38 HOXB cluster, yeah, HOXB cluster.gtf. That's what that file's called. Let me try to read it in and let's see if it works. I think that worked. It looks like an error, but it's actually not. Because we can come over here and, yep, we can look at it. So that got the data correct. We have chromosome 17. We have all this correct. Fantastic. And then let's go ahead and read in this one, mm39 HOXB cluster.gtf. And that should also work. MM39. Oh, I accidentally didn't change the name here. Let me go ahead and redo that. MM39. In fact, let's do it. Mouse MM39 HG ano. Mouse MM39 HG ano. That worked. Awesome. So let me go ahead and change all instances of the dog one to the correct mouse one. Okay, cool. And then dog to, let's name it something useful. And dog MM39 Chrome 11 ano. Okay, cool. That will work. So now that we've read in our GTF files, we want to actually extract the data in meaningful ways for our plot. Because if we go back to our plot, notice how the genomic region isn't necessarily showing you the entire chromosome or anything. It's literally showing you the slice that we extracted. But to make sure that R knows to actually compare the relative sites instead of like the absolute sites, we need to make it relativized to the location on each chromosome. What I mean by that is we literally need to take the values of the genes that are the absolute locations on chromosome 11 and chromosome 17 and relativize them to this 100,000 base pair window. The way we do that is to tell it where our start and end locations for our regions are, and then get the relative location of each gene on this segment of the overall chromosome. So to do that, we're going to edit these values here, which were boilerplate from previous start and end locations. And we want to make sure that we're getting all the data correct. So we're going to, let me change this actually, so that instead of human chromosome 17, it's literally chromosome, yeah, instead of chromosome 7, it's chromosome 17. So let me replace all those. And then let me replace, yeah, no, that's actually correct now. Okay, so now that that's correct, I need to specify a few other things. So it's, let me actually explain what's going on. So this code is going to look at our gene GTF data frame and extract data like the gene name, start and end locations, chromosome name. So it's going to take the gene name data from X9 and actually get the gene name in quotations. We can see that here. And then it's going to look at chromosome 17. So we need to specify this as 17. And then we need to feed it the start and end location of our actual setup. So how do we get that? Well, we actually had that in the file that we used to get SAMtools FadeX. So we're going to feed it this start location. And it doesn't want commas, so we'll get rid of the commas. And then this end location. And we can specify, or we can verify that these are valid, because all of our human gene annotations need to be within this range, and these all look to be within that range, which is perfect. And we also need to make sure that these, and we can verify it, like, yes, notice how these three are the same value, these all need to stay the same value. So we can do, let's do Control-Shift-Z. And now we'll just copy this value over here. And then that should work the correct way. Let's try it. Could not find function arrange. Interesting. Could not find function arrange. Arrange. Could not find function arrange. There must be some library that's not fed into this. Let me try loading all the libraries real quick. Okay, let's try that again. It might still not work. Let's see. Oh, that did work. So I guess one of the libraries was not working. Let's go ahead and open this setup now. Human chromosome 17 ano. Okay, cool. That is perfect because now we have extracted all the data from our GTF file that we want. Humans and chromosome 17. And perfect. Now, oh, and yeah, let me say too. Notice these are now relativized. Because if we look at the actual absolute location of HOXB1, it is from 48 million, 5 blah blah to this, whereas the relative location on this 100,000 base pair window is 1719 to 4,202. Perfect, so it worked great. We're gonna do the same thing for mouse. So we'll double check that the location of the chromosome is correct. It's not because it's actually chromosome 11. Here it says 16. So we'll edit that, chromosome 11. We will get the start and end locations from our fadex command. So we'll go ahead and write down that for start and then this for end. And we'll copy the start down here as well. And let's try running this one. Let's see if it worked okay. Worked almost good. Let's see. So we have HOXB 9876. It looks like 5 did not work. So there has to be an issue with HOXB 5. What is the issue here? Let's see. HOXB 5 did not work. 1, 2, 3, 4, 5. So it looks like the genomic locations are valid. And there has to be an issue. Ah, I see. I accidentally typed gene name instead of gene name. So I'm going to edit the GTF file real quick. That's actually a good example. I didn't even plan that, but I'm actually glad it happened because it helps us show the process of diagnosing an issue like that. So that should be saved. We will now go ahead and read the GTF file. And then that should work. Yeah, so now that's corrected. Now when we try running this, it should capture the gene name correctly. Let's check it out. Yeah, so that worked. Okay, awesome. Now that all that worked, we'll go ahead and use this to extract again the data we want into a specific setup for SV-BioI called genomic ranges. So we'll go ahead and read that. So this is going to be HOXB cluster ano. Let me double check. So HOXB cluster ano. Yeah, okay, so let's bind these rows together and we'll check that that worked because HOXB cluster ano now should have the genes for both oh yeah, actually I should have caught that. This has to be mouse. And this is not correct either. So let me go ahead and fix both of those. So this should be human chromosome 17. This should be mouse chromosome 11, the same value as up here. So let me go ahead and do that. Again, glad we're caching these things. Let me rerun all that stuff. Now let's take another look. Let's take another look at that. Let's see, HOXB cluster ano. Okay, now it's working. We have the correct location, mouse instead of dog. Good. Okay, now let's try doing the genomic ranges on that. That should give us formal class genomic ranges. And we should also see metadata like the gene names. Let's see if that did work. Gene. Okay, gene name. Good. So that did work. Okay, so let's try remaking this plot because nothing's going to change with this plot. We just want to make sure it didn't get broken. And it looks like it didn't, which is great. I know nothing happened, but we can like double check by plotting black and then going back to our blue. Yeah, perfect. So let's go back to our blue setup. Okay, cool. Now we're going to try adding the gene annotations that we just created after all that work. Let's see if that worked okay. This is basically going to try adding a rectangle for each of the gene locations. Let's see if it works. So we're going to do this. There's no errors. Okay, that's awesome because that seems to have worked pretty good. It looks like there might be an issue with human chromosome something. So we don't know what that one is. To figure out what that is, we're going to try adding a gene name label. So let's see if it actually works. Okay, this worked really good. So we added the actual gene names to each of them. And we can see, just like we saw in IGV, in human, HOXB, the HOXB cluster goes from left to right, whereas in mouse, it goes from right to left. So we have HOXB 1, 2, 3, 4, 5, 6, 7, 8, 9. In mouse, it goes HOXB 1, 2, 3, 4, 5, 6, 7, 8, 9. I do think there's an issue with HOXB 3 and 4. Let me go back to IGV and just revisualize. Let me see if that is reflective of how it actually looks. I think it's not. But let's see, HOXB. Oh, actually, that is correct. So we can look. HOXB 3 and 4 do overlap in the human genome. So actually, this plot is correct here. I am going to make the labels a little bit smaller. To do that, I can illuminate how the layers are set up in our ggplot object. To manipulate the text, we would want to go down here to the geom text layer, which is layer 4. Let me try, as an example, changing the size of the text to 1.5. It's going to be too small, but at least it is working. So we're going to try 2, and we'll just keep trying till it looks okay. 2 is all right. 4 might be better. It might be too big. I'm not sure. 4 is just apparently, I guess, default. Let's try 3. That might be a good middle ground. I think 3 looks pretty good. It's not perfect. Like HOXB 8 and 7, 6 and 5 in mouse are pretty close, but it's pretty good. I'll try one more, 2.8. And this is just raw, like quick trying it. I think this is pretty good though, because I think it's enough to differentiate, for example, 7 and 8 while still being legible. I think if we really wanted to, we could do 2.6 or something, and that would help a little bit more. I think that actually, yeah, that looks pretty good. Last things we'll do, we will go ahead and save a plot without any, what's it called? Yeah, without any of the annotations. Or actually, no, I'm just making stuff up. What I mean is we'll save this plot as is without an axis title and plot title. Then we will go ahead and add titles and then be done. So we're going to go ahead and save that. And we're going to do height like 6 or something. I think that might look pretty good. We're going to go ahead and save that to our current directory like this. Let's go ahead and try opening that. It should be a PDF file. Now we can go date modified. Okay, that's the plot. That looks pretty good like that. The last thing we'll do is we will go ahead and add a plot title, and then I think that will be good. So we're gonna do HOXB cluster Synteny and human issue 38. and mouse MM39. Human cluster, Synteny, and human and mouse. And then this is gonna be HOXB cluster locus size. Because it's not necessarily genomic position from the start of chromosome 11 to the end, et cetera, et cetera. It's really locus size and relative location. And then we're gonna do color. No, that's fine actually. Yep, that's fine. So we're gonna get that out. Let's see if that works the way we want. In fact, we'll move this up here. Okay, let's go ahead and check that out. Okay, that looks pretty good. In fact, let me just see if this works real quick. No, that didn't work, which is fine. Okay, I was just gonna try to adjust that title. But this is looking really good because we have our chart title now and we have the HOXB cluster locus size. In fact, I'll just make that uppercase. Feel free to adjust the colors and stuff as you would like. It's up to you how you want to do it. I think that looks good. For this one, for fun, on that note, I will make this a different color. I'll just do like, I don't know, maybe orange, red might look good. Could try it. It doesn't look actually that good. Maybe purple. Purple's all right. Purple's all right. I'll try one more. I'll try orange. Yeah, I kind of like the orange. Maybe a darker version. No, that's actually not that dark. It's up to you. You have free will. I'm just playing around with some stuff. But I think this might look... I kind of like that one. All right, we're going to run all these again. And then we'll go ahead and view the plot. Man, you know what? Man, I like the blue. I like the blue, actually. So I'll just put like this one. No, not as good. This is optional. You don't have to do all this stuff. Well, that's fine. Yeah, we'll just go back here. And then let me go ahead and change the title of our plot to something informative, like same name, but like with title or something. And then you can adjust the height and stuff. I'll do 7 this time just to differentiate. And we know it works because we can literally restart RStudio and we will try running all this code and it should all work in one chunk. So I'm gonna go ahead and just save that window and stuff. We're gonna try reopening RStudio. And the reason I did that is because I want to illustrate that now that we've edited all the code, we should be able to just run the whole script in one go. And a lot of the variables, in fact, let me go ahead and clean all these variables up. So we're starting fresh with this script. But we have all the files ready to go. We're going to run everything. And it should give us the plot, and it should save it to our computer. So we can go ahead and go to date modified. This should open it, and then it worked great. In fact, we can go ahead and full screen this. So after that work, this is our result. We can see the HOXB cluster Synteny in human and mouse. We have a lot of alignment for these genes. Basically, the whole cluster is conserved, and it's just flipped. The alignment direction is negative, so we can literally see this flip right here. HOXB 9 over here, flipped over here. HOXB 1 flipped over here. Same with these setups. So it's really cool. There's a lot more you can do with this plot, but I think this is a really good stopping point for us today. Hope you learned a lot. All the code for today and all the files that I generated are going to be stored below on the GitHub for this project. Play around with it. Hope you learned some cool stuff. Keep digging. It's cool. You can do this with literally any cluster you want, with any species you want. Have fun.